Okay, we're moving into section 4, getting into how we get from code to stuff. And since I was talking about graduate school last chapter, I've got something up here, graduate school. Okay, this is it. I'm gonna graduate. No ifs, buts, or maybes. The how, when, and why would be nice though, is that you get a degree in FM when you have the right number of credits for the right stuff. You get a bachelor's when you get the right number of credits for the right stuff. In science graduate school, you get the degree when a committee decides that it's ready to grant you the degree based on your research and your write-ups and your presentations to the committee. There's a certain number of graduate credits, classes that you need to take, but it's a much more wide open, iffy gray area kind of what do you need to do to get all this stuff together. And there's a number of hurdles that you need to get over that is that one of the exams that I took with a committee, I think the week before the exam, as I talked to my main professor, the professor who supervised the lab that I worked in, said, so what do you want me to know? You went, oh, you got to know a lot of history of science. It's like, what? Okay, this is news. That wasn't really my concentration for. It is now. I teach based on history, but a lot of that had to do with him going, oh yeah, you got to know all that stuff, and he was right. Is that, to me at least, building stuff through the history makes it more graspable how all of this complex stuff was kind of put together. Bayesian approach, where you kind of build on what you know and you build more stuff and you build more stuff and you build more stuff, and it makes more sense and makes more sense and makes more sense. And that's what we're going to do with this chapter on genetics. It actually makes sense to start with that cartoon. And to start with Mendel, who we've talked about already, who, because of his particular circumstances, he was working in a produce-producing monastery, and they needed him to grow pea plants, is that he was very interested in how certain features that seemed to only come in either or varieties were generated, genetics, in these pea plants. And he taught himself how to pollinate plants and restrict pollination so that when he crossed this plant with that plant, he knew that's what he was doing. He knew that the pollen contribution was coming from one plant and the egg cell, which is in the base of the flower, contribution was coming from another plant. And he started to cross these discrete things and found out that whatever was making the traits happen, whatever was generating the traits, those he called the genes. But when you got different variants of the same trait, he decided those were produced by alleles. Now, he had no idea what was happening inside. He was just seeing the patterns. And as a way in, he was looking at the most simplistic patterns. A pattern where if you have one allele making one trait, in certain combinations, it could hide the presence of another allele making a variation of that same trait. Tall could hide short. Purple flowers could hide white flowers. And the only way to get white flowers was to have the two alleles together and no purple to cover it up. The only way to get short was to have two alleles together and no tall to cover it up. And so he came up with the idea of dominant alleles and recessive alleles. Dominant alleles could hide the presence of a recessive allele. Now, of course, what we now know is that what he called a gene, we refer to a DNA code for a particular type of protein. Now, in those codes, there are variations, and those variations are alleles. Sometimes the alleles don't really do anything in terms of changing the protein that's being coded for. And sometimes they do significant things about the protein it's being coded for. Typically, a recessive allele is a code for the protein that produces a non-working version of the protein. So if you have a working version of one of your pairs and a non-working version, the working version makes the trait happen. And it hides what happens when there's no working version. The base color of a pea flower is white. If you have no allele that can make a purple pigment, you get a white flower. If you have one allele that makes purple and one that doesn't make anything, you still get a purple flower. The tall short was a gene for growth hormone in pea plants. The recessive was a growth hormone, it didn't do anything. Is that those plants grew, but they were stunted. All you needed was one functional growth hormone code to make enough growth hormone to get a normal height. So what he considered tall and short, we now consider normal and stunted. And it does turn out that a tremendous number of recessive diseases, well, actually genetic diseases, are not always, but they very typically are recessive because they are the product of codes for a protein, and that version, those alleles don't produce a working protein. So whatever that protein was doing isn't happening. And so you get a disease from the fact that you have a non-working protein doing something not so important that it kills you right off the bat, because then we don't know you're there, but it affects you through your lifetime. Maybe it kills you eventually. So he published a lot of stuff. He developed the way that these things are written down, where alleles are represented with letters. And in his case, since he was working dominant recessive, a fairly simplistic form, capital letters for dominant, lowercase of the same letter for recessive. You pick the letter various ways, usually on the trait, but you run out of letters eventually. And you can use other kinds of symbols. Dominant recessive doesn't work. I mean, he had a handful of traits in pea plants that were dominant recessive. Pea plants have lots and lots and lots of traits, only a handful are dominant recessive. And if you think about it, that makes sense, is that you're not going to get that many mutant alleles that produce a non-working version of the protein, where if you get two of them together, it doesn't just eliminate the embryo and you never see the pea plant. So things that are expressed, same thing's true for, I mean, in humans, there's dominant recessive traits. And again, they tend to be kind of superficial traits, like your hairline or your eye color. Things that if you've got two non-working versions, in the case of eye color, you can see the blue layer shine through because you're not making dark pigment. But a lot of things, two non-working versions would be bad for you. So Mendel, though he worked with dominant and recessive, he developed a lot of standards for how do you write this stuff down? How do you present crosses between parents? How do you present the alleles? How do you present the mixtures? As I talked about last semester when we introduced genetics just as a general concept, nobody really paid much attention to Mendel. His papers tended to be full of math, and biologists still are not crazy about math. Back then, naturalists, as they were called, were the assumption is they were interested in Mendel's papers, but too confused by the math to really build on them. It took a couple of decades of better math training in naturalists for people to kind of come back and rediscover Mendel's work after Mendel was dead and use it as the basis for the bottom of genetic study. Mendel developed a bunch of rules based on his pea plants. One of the rules was the rule of independent assortment, is that he looked at a bunch of different features that came dominant recessive, and the dominants didn't go together, the recessives didn't go together, everything sorted total independent from one another. We now know that's because every one of his genes were on separate pieces of DNA. They were not linked together. They were not physically connected to one another. As people started to build on Mendel's work, and they started to reproduce similar studies in their own study groups, their own study organisms, they found that that wasn't always true, that certain genes tend to travel as packages, that a dominant and recessive allele might seem to be somehow linked together and always go on together in this particular family line, whereas in another family line, the two recessives were always together. Another family line, the two dominants were always together. And they discovered in the early 20th century linkage. Now, in organisms that use unmatched chromosomes for sexual differentiation, like we do, they discovered eventually a very specific type of linkage called sex linkage. That was a big clue as to what was going on, is that in sex linkage, now I have only one X chromosome. So any code on that produces a non-working protein, I'm kind of in trouble because I don't have a second one to produce a working protein and cover it up. So genetic diseases that are sex-linked tend to appear much more in the unmatched version. In birds, it would be different because the unmatched are the females. But in mammals, a very big study group, mice and people and all sorts of things, they knew that there was a mismatched chromosome in the males and a matched chromosome in the females. They knew that there were genetic diseases that would show up way more in males that would rarely but occasionally show up in the females. And they did the math and discovered, okay, these things have got to be on these chromosomes. They're connected together by being on the X chromosome. And females do have two, so they're much more likely to have a dominant hiding a recessive disease allele. Males don't have that possibility. They only got one X. Anything on there, that's their only code. So linkage also associated with sex linkage. You do enough of this, and you eventually discover you can't always depend on the linkage. Sometimes the linkage breaks. Things that were connected somehow swap off. And what we now know is crossing over. And as they eventually discovered that the chromosomes actually were holding the genes, because that's what linkage is, genes on the same chromosome, that the rate of separation could be used to map out where the genes were on a particular chromosome. If here's a gene and here's another gene on the same chromosome, there's a lot of places in between where that linkage could be swapped off by crossing over to the other homologous chromosome. Whereas if the two genes are right together, the odds of those separating into, because of a crossing over event, a lot smaller. By mapping out the rates, you could figure out where these things, how close together they were. And there's only so much room on the chromosome. So you start to figure out if here they're getting out close to the tips. And so something else that's pretty close is probably between rather than beyond. You can map it with another one and make sure that that's true. And if you take a genetics class, lots and lots of math, you do the math in order to figure all this stuff out. And if you're going to be a biology major, you're taking a genetics class. There's no way around it. Is that I know at SUNY Albany, when my daughter transferred there, we didn't have enough students to actually put together a genetics class. And so She transferred in as a junior because she'd gotten her first 2 years worth of classes here, except she couldn't take anything because everything at the junior level required genetics as a prerequisite, and she wasn't able to get it here. She had to take it over the summer before she actually started as a full-time student in the fall. That happens. Something associated with this that I'm just going to throw in here. It's just the basic idea of chromosomes. There's thousands and thousands and thousands and thousands of genes. Cells, to make more cells, they want to make sure that a complete copy of all the codes goes to the new cells. You got to make copies, and then you got to get the copies distributed, right? We talked about that in mitosis. And we talked about chromosome number, right? Is that how many pieces do you need to make copies of and move to get them separated properly? A low number, fairly easy to do and not mess it up. A high number, sometimes they don't go where they're supposed to go. The more chromosomes you have, the more errors you're going to make during divisions, where you've got 2 chromosomes to go, but they both go one way. For R46, that happens relatively commonly, or there wouldn't be something called Down syndrome. Down syndrome is the only example of bad separation, except for the sex chromosomes, the only example of bad separation during meiosis that a human embryo will survive long enough so that you know it happened. Is that in most other cases, the embryo dies early on. You may or may not even wind up with it testing for a pregnancy test. And again, reasons we'll talk about later. The flip side of that is the more you have chopped your DNA up into pieces, right? Instead of one long chromosome chopped into pieces, the more different ways there are to sort through them when you're making offspring, right? If you only have 2 chromosomes, all you can send on to your offspring is 1. All they're getting from the other parent is 1. And there's not a lot of ways you can mix those together. 2 chromosomes, there's a couple you can have more mixtures. 4 chromosomes, more mixtures. Is that 46, 23, 23, the number of ways you can sort through those. You can do the numbers, but there's a really big number of possible mixes. So variation, which is a huge driver of evolution, is increased with more chromosomes. So higher numbers, better variation, more screw-ups in cell division. Low numbers, fewer screw-ups, less variation. There's another secret here. If you have a very low number of chromosomes and you're reproducing by yourself, which a lot of animals are capable of doing, and plants for that matter, then if you have a low number, so you split them up and then you recombine them. If you have a low number, you have some decent odds of actually producing genetically identical offspring to yourself. They got the same chromosome mix that you started with. And so in low chromosome numbers of self-fertilizing organisms, you get a kind of a quasi-asexual reproduction and the advantage of making genetic copies that entails. So I will say generally that's not talked about, but if you do the math, it's pretty obvious that it works that way. Okay, I'm going to stop there.